Okay, so welcome to the Environmental Research webinar. So this is our first webinar in winter 2025. And, today is again, the new Lunar Year Eve. So for those who are celebrating Lunar Year, so congratulations and then Happy New Year. I am Sungkyun Park, faculty member of Epidemiology and Environmental Health Sciences. So this seminar series is sponsored by the Michigan Life Stage Environmental Exposures and Disease Center. Before I introduce today's speaker, so please note that please use the Q&A on the bottom to enter your question, and then we will cover your question after the talk. I'm very excited to introduce today's speaker, Dr. Bambarendage Perera, also known as Pini. So Dr. Perera is a research assistant professor of environmental sciences here at the University of Michigan School of Public Health. Dr. Perera earned a Bachelor of Science from the University of New Orleans and her PhD in biological sciences from Louisiana State University. She also completed postdoctoral training here at UM under the guidance of Dr. Dana Dolinoy. Dr. Perera's research is centered on understanding how environmental exposures, particularly during critical developmental stages, impact gene expression and health outcomes through epigenetic modifications. Her work focuses on the mechanisms of genomic imprinting and non-coding RNA regulation, utilizing the mouse as a model organism. Dr. Perera has been recognized with awards such as the Environmental Mutagenesis and Genomics Society Young Scientist Award. Today, Dr. Perera will speak about developmental effects of lead exposure, alterations in placental and embryo weight, and imprinted gene regulation. Please join me in welcoming Dr. Perera.
Good afternoon, everyone, and thank you, Dr. Park, for that kind introduction. And I also want to take this opportunity to thank M-LEEaD for giving me this opportunity to share our most recent work about this gestational lead exposure effects in the mouse placenta and embryo, and I hope you enjoy the talk. So this is a very quick outline and an overview for the presentation so that you're able to follow through. And feel free to put your questions in the chat and I'll try to answer them as they come. We will first go through a brief introduction to epigenetics, followed by genomic imprinting, and I will then talk about some previous data on published work from last year on perinatal lead exposure effects, and then move on to our current work for the in utero effects of the placenta and then the embryo. And finally, I will wrap up and end with conclusions and future directions, and then open for questions. According to the Developmental Origins of Health and Disease, also known as the DOHAD hypothesis, exposure to environmental toxicants can start as early as in utero development and even before before gestation too, and can have long-lasting health effects until adulthood and throughout the life course. So my current research program aims at understanding how early life exposures, like lead, impact long-term health and disease risk. And my research is specifically focused on how the epigenome can contribute to this disease risk. And there's like epigenetic dysregulation, like what are the mechanisms that contributes to later life disease? So what is epigenetics? Epigenetics refers to the changes in gene expression that occur without altering the underlying DNA sequence, which could be composed of modifications such as DNA methylation, histone modifications, and non-coding RNA. The epigenome is dynamic, and these changes are reversible. So it's highly influenced by the environment, be it exposure to chemicals, diet, or exercise. So as we know more about how exposures can impact gene regulation and long-term health, we can find ways to either identify diseases early through epigenetic biomarkers that inform some of this later, that predict later life disease outcomes. And as we know about how these epigenetic mechanisms are dysregulated by environmental exposures, we can identify we could identify modes of intervention through diet or molecular mechanisms like non-coding RNA. Switching gears to talking about lead effects and its sources, there are several different sources of lead exposure, with the most prominent ones being contaminated soil and water. And us being in Michigan, we're no stranger to lead exposure as a problem. And it is a very well-known neurotoxicant that affects childhood health and particularly neurodevelopment, even though there are other tissues that are impacted and other long-term health complications as well. Previous literature in humans suggests that lead exposure is linked to dysregulated DNA methylation and also transposon regulation, so basic genomic stability. In mice, perinatal lead exposure is linked to tissue and sex-specific DNA methylation changes, and we will touch on one of these examples in today's talk very briefly. When we talk about epigenetics, I always have to talk about these like two resetting events that happen during development. So in the top right here is showing the mouse development, kind of the stages through the mouse development. DPC stands for days post-coitum, so this is before birth. And then the y-axis here is kind of showing the DNA methylation level. So the top right here is most methylated, and the bottom right here is least methylated. So in mouse development, there are two critical epigenetic resetting events that occur. First happens during germ cell development, erasing and resetting these epigenetic marks to make sure that there's genetic integrity for future generations. And I've highlighted this 13 to 15 DPC time point because this is the time point that is used in this gestational developmental study that I will be talking about today. And the second occurs right after fertilization, reprogramming the zygote to support for embryogenesis. And these processes allow for proper growth and development both for the placenta and the embryo. And one important point to note here is that imprinted genes and these like IAP particles kind of skip this second resetting event that occur. So these two stages are therefore very critical windows of susceptibility for environmental exposures and stressors. So that's kind of highlighting the significance and importance for studying pregnancy time point, and specifically these two time points in terms of epigenetic resetting. So what is genomic imprinting? So in sexually reproducing organisms, genes are typically expressed from two parental alleles, one from mom and one from dad. However, because of genomic imprinting, a subset of these genes are exclusively expressed either from the mom or the dad. So by definition, imprinted genes are monoallelic gene expression based on its parental origin. And this is just a couple of images just to show the evolution of imprinted genes. Imprinted genes are highly conserved in eutherian mammals. So eutherian mammals are mammals that have a functional placenta. So in the image to your left, we have monotremes and marsupials, the egg-laying mammals, and the non-placental mammals. You can kind of see that there are very few imprinted genes evolutionally conserved there, but a lot of these imprinted genes were discovered and are highly functional in placental mammals. And also note, imprinted genes, they're either maternally expressed or paternally expressed, so we call them MEGs or PEGs. But besides their MEG or PEG name, they also have multiple other names. So when you're working on genomic imprinting, when you're reading the literature, you kind of have to dig a deeper dive into the literature search when you're working on imprinted genes. So a quick overview on imprinted genes. Historically, it was first discovered in the 1980s through nuclear transfer experiments, where it was shown that you need both maternal and the paternal genomes to produce a successful offspring. So to see whether if maternal and paternal there's no specific contribution, then having two paternal genomes should give rise to a normal offspring, or having two maternal genomes should be able to give rise to a normal functional offspring. But these experiments showed that two paternal genomes or two maternal genomes is basically embryonic lethal, meaning that you need a balance of both maternal and paternal genomes to have a successful life. For instance, here is a very brief overview of how imprinted genes work. It is a complex process, but I've kind of simplified it here for you. Here I've shown, and there's like closed circles showing DNA methylation, and these arrow kind of indicating active transcription. So here you have M standing for maternal allele, P standing for the paternal allele for a mom and a dad. So this is an example for a paternally expressed gene that is maternally methylated or maternally imprinted. During epigenetic resetting that I mentioned before, at oogenesis and spermatogenesis, a mother's alleles get completely methylated while the father's alleles get completely demethylated, which ultimately gives rise to an offspring or a zygote that contains a paternally expressed gene that is maternally methylated, similar to its parents. So this is kind of how that imprinting mark passes from one generation to another. Maternally expressed genes kind of have the opposite way, vice versa. They have the maternal allele expressed and the paternal allele imprinted or methylated. So they're technically called MEGs. And I've given a couple of examples here for a maternally expressed gene and a paternally expressed gene, and we'll revisit in the talk later on. And these are very highly expressed in the placenta and embryo and throughout pregnancy and development and throughout postnatal development as well. And I'm particularly interested in looking at its functions in the brain because it is also important for hormone regulation as well. So why are imprinted genes important? There are a myriad of problems that occur with dysregulated imprinting or loss of imprinting. They are very critical for growth and development, specifically during pregnancy. And here there are a bunch of acronyms here, but just to emphasize that the placental development, uterine development, imprinted genes play a very critical role. An example of a disease phenotype, a syndrome associated with it is like BWS, which is Beckwith-Wiedemann syndrome. And then there are genes that's kind of associated, imprinted genes that's associated with that as well. And not only pregnancy, but this can feed on to, this can persist through postnatal development and maternal caring behavior and hormone regulation as well. So imprinted genes are important. If it is dysregulated, it can kind of dysregulate multiple gene pathways throughout the life course. And a couple of other diseases here is Angelman syndrome and Prader-Willi syndrome, AS and PWS. So that is the example that I will very briefly touch here. This is a very complex diagram of what the genomic locus looks like, but I will kind of walk you through the main points. So this is a case where the imprinted gene, there are multiple imprinted genes in one locus. So in green, you're seeing non-imprinted genes. In blue, you're seeing paternally expressed genes. Red shows the maternally expressed genes. So here, any deletion or loss of imprinting for this blue region will result in Prader-Willi syndrome. And similarly, if there's any deletion or imprinting, loss of imprinting in this red region, will lead to Angelman syndrome. And interestingly, these two syndromes, phenotypically, the disease phenotypes are complementary. I have not put any pictures just because I am uncomfortable sharing human pictures for these slides, but you can look them up too. And I like to highlight 3 specific imprinted genes that's contributing to these two disease phenotypes. One is MAGL2 and SNRPN, which is the main contributor for Prader-Willi syndrome, and what's called UBE3A, which is the main contributor for Angelman syndrome. With that, I will transition to talking about some of this preliminary data and a paper published by Dr. Wang and Morgan last year. This is a part of the study that was conducted through the NIEHS Target 2 program, where we generated perinatal lead exposure mouse models demonstrating persisting epigenetic changes throughout the life course. So we used a mouse model that were about 2 months old virgin females that were exposed to either control or lead-spiked water from 2 weeks prior to mating, all the way through mating, birth, and weaning. And the offspring were switched to control water after weaning, and they remained so until 5 months of age. We used 1 female and 1 male per litter for 6 litters, and they were used to profile their DNA methylation from 3 specific tissues for this study: blood, cortex, and liver. And here are kind of the main findings from this previous work. This is showing the goal analysis or pathways analysis for differentially expressed DNA methylation. So this was done through whole genome bisulfite sequencing to kind of identify genome-wide DNA methylation differences. So on top here, F stands for female and M stands for male. There's blood, cortex, and liver, and kind of showing the different different pathways that are impacted by lead exposure for DNA methylation. And it shows that more biological pathways and more biological processes are impacted. And if we switch from the same study to looking at imprinted genes, using the brain tissue, this is the data for the cortex, and kind of weeded out the imprinted genes. It shows that the genome-wide DNA methylation shows that there's very sex and tissue, because this is the cortex, specific DNA methylation at imprinted loci upon perinatal exposure to lead. And it shows differentially methylated regions, which are called DMRs, at what we call imprinting control regions, or ICRs. These lead-induced DNA methylation changes at DNA methylation changes at imprinted genes persist beyond exposure, with sex specificity. If you look at MEST gene right here, it shows that the DNA methylation has decreased or hypomethylated, specifically in males, while the NNAT imprinted gene is hypomethylated in females specifically. So there's like sex specificity going on here because of lead exposure. So in summary, imprinted genes are susceptible to exposures during development, especially during those 2 epigenetic resetting events. Exposures during pregnancy can dysregulate imprinting, and these changes may occur at DMRs, differentially methylated regions, or imprinting control regions, and that these epigenetic changes may lead to disease phenotypes later in life. So this leads to our question that's central to our current study. Are imprinted genes changed upon lead exposure in the placenta and embryo? Because they are very highly expressed in these tissues. So our question directly asks about these direct impacts of lead at pregnancy. So the mouse model used for this study is similar to the perinatal exposure model because we used a similar dosage of lead and then started the study design exposure 2 weeks prior to mating, similar to our previous study. However, the exposure only continued through pregnancy in this study design, where the placenta and the embryo tissues were collected at 13 to 15 days postcoitum. These animals were time-mated, meaning males were put into the female cages only for the duration of 2 nights. That's why we have a 2-day window for this study. So it's very highly tightly controlled. And I've shown in the image, there's like pink marks that shows exactly how I cut the tissues for the rest of the study. So it's kind of like a sagittal section for the placenta and kind of looking at the embryo head. Sadly, I can't dissect out the brain from these, the embryos. They're too small. So we basically dissected the embryo head to kind of suggest what's going on in the embryonic brain. So here is kind of the overall results for the observations that we saw. When we look at the litter distributions and the viability of the embryos upon in utero exposure, we see no significant differences between litter sizes that are that range from 9 to 10, 9 control and like 10 lead exposed litters, which is shown here. So there's like similar litter distribution. We also see a similar number of total viable embryos for both of these groups. And we observe no significant differences between males and female embryos. So they are both born, they are both viable. So no major issues there. But now we'll get to the fun part of the talk, where we kind of tease apart whether lead exposure impacts the developing placenta, and then we will touch base on the embryo data. So here, for the next slide, I have shown raw data collected from the 9 control and the 10 lead exposed placenta weights. So this plot shows that the placenta weights over these different litters by exposure groups, showing control in yellow and lead in blue, that the lead exposed litters trend towards a higher placenta weight relative to control. And this is significant. The weight differences between control, yellow, and lead in blue is significant controlling for litter effects. And these weight differences were statistically different when stratified by sex, with females shown in purple and then like males shown in gray. Again, controlling for litter effects here as well. And we isolated RNA from the placenta, similar to the previous study, 1 male and 1 female per litter for each of these exposure groups, and performed RNA-seq to assess differentially expressed genes, which I will refer to as DEGs from now on, imprinted genes called IGs from now on, and pathway analysis. For today's talk, I'll only be focusing on our data assessing imprinted genes. And we ended up with 8 to 10 samples per sex per group for this study. So these are the N numbers used for the rest of the talk for the placenta. And for the remaining slides, I'll also stick to these colors shown to distinguish our data by exposure groups, shown in blue, that compares control versus lead. As well as our 6 stratified results shown by purple for females and gray for males. Our sequencing reads were first pseudo-aligned to the mouse genome, which gave us a little over 26,000 genes, I guess close to 27,000 genes. And after filtering and normalizing our reads using EdgeR, we ended up with 14,287 total genes detected from the placenta. From that, we were able to see over 4,000 DEGs by exposure group, with over 3,000 for females and about 849 for DEGs for males. And then we filtered these DEGs based on a list of 300 canonical and non-canonical imprinted genes. So these gametic imprints and non-gametic imprints based on previous publications, and also looking at this gene imprint database. After filtering based on these list of 300 known list of genes, we were able to successfully detect 151 of these in our RNA-seq DEGs. And the next few slides will show volcano plots for the magnitude of changes seen from these results, and kind of breaking it up apart from combined sex for comparing the two exposure groups, and then females and then males. Starting with exposure group, the imprinted gene expression in the placenta showed a total of 58 genes that were significantly changed relative to control. And 40 of these changed genes were more upregulated with exposure, and then 18 were downregulated with exposures. And among these top hits, GRB10, which was a gene that was shown in a perinatal lead exposure study, and SNRPN, which is linked to Prader-Willi syndrome, and then DO3 is involved in thyroid hormone inactivation. So kind of shows this hormonal regulation and then general placenta development that's kind of linked to some of these general dysregulation of imprinted genes. For females, we see 38 genes that were significantly changed, 38 imprinted genes that were significantly changed relative to control. 24 of these were increased and 14 were decreased by exposure group. And of note, this PHLDA2 gene is the gene that's linked to Beckwith-Wiedemann syndrome that I mentioned before. And ASCL2 is also an imprinted gene that is important for trophoblast function. So those are kind of insinuating why these are important for placental development. For males, we see 13 imprinted genes that were significantly changed relative to control. 11 of them were more increased and 2 were decreased by exposure. And again, ASCL2, I kind of mentioned what the function is, the trophoblast function. PLAGL1 is also known as ZAC1, is linked to cell growth, and this can be linked to this like increased placental weight that we have been seeing for the placenta. So overall, here's a distribution of our differentially expressed imprinted genes by exposure group and our sex stratified results. And among these nine imprinted genes that were found in all three groups, gametic imprints like DLK1, GRB10, SNRPN, and ASCL2 are particularly sensitive to lead exposure in the placenta. And I'm also showing exist here because that is important for X chromosome inactivation in females. And I've also put arrows here for SNRPN and MAGL2. Both of those genes are the ones that I highlighted when we talked about the Prader-Willi syndrome contribution. So now we'll switch gears and look at our embryo data and embryo head data. This is the raw data plot that shows the overall embryo weight distribution for different litters by exposure groups, showing control, again, in yellow and lead in blue. The lead-exposed litters, again, trend towards a higher embryonic weight relative to control. And it's also interesting to see that the embryo weights cluster very well together among litter mates compared to our placenta distribution, which was more variable. The embryo weight difference between control in yellow and lead in blue is statistically significant, controlling for litter effects, very similar to our placenta data. And these weight differences were statistically different even when it's stratified by sex. And similar to the placenta, we performed RNA-seq here we use the embryonic head, using 1 female and 1 male per litter for each group. And we ended up with 9 to 10, which is the maximum we could get, 9 to 10 samples per sex per group for the analysis. And for after alignment, we found, again, over 26,000 genes that were sequenced. And after filtering and normalizing, we were able to detect about 15,000 total genes in the embryo head. And among these, about 2,662 DEGs were found by different by exposure group. And over 600 were contributing from females and 700 were contributing from males. And after filtering for our imprinted gene list, we were able to successfully detect 161 IGs from the embryo head. And we'll go through our plots for the next 3 slides. And here, starting with the exposure group, the imprinted gene expression in the embryo head showed 23 genes that were significantly changed by lead, and 21 of these were increased and 2 were decreased. And among the top hits, there's SLC22A2. This is a gene that is also located in the same genomic region as the IGF-2 receptor and ARN imprinted regions, and is very highly expressed in the embryo and the brain. And I'm also showing RAS-GRF1. That is a RAS protein-specific guanine nucleotide releasing factor that is important for long-term memory, which can interestingly, they're also regulated by a non-coding RNA called Pini interacting RNA, which I'm coincidentally also working on. So that's very particularly exciting to see. Well, it's bad for the animals, but kind of exciting to see the data linking these complex regulatory networks for imprinted genes. For females, we see 9 genes that was significantly, all of them were increased relative to control. And those 2 genes, RAS-GRF and SLC22A2, popped out here as well. For males, we see 10 genes that were significantly changed. 3 of them were increased, 7 were decreased by exposure. And interestingly here, these are males. EXIST is typically not expressed in males. And we do see EXIST being very highly expressed only among our lead-exposed embryo heads. This will definitely need to take a closer look at the DNA methylation assessment of these specific embryo heads to kind of figure out mechanistically what's really going on in these male embryo heads. So overall, here's a distribution of the differentially expressed imprinted genes for the embryo head. Among the 2 genes that are found in all 3 groups, here they are, all 3 groups, gametic imprints like RAS-GRF1 is particularly sensitive to lead in the embryo head with functional implications for long-term memory and neuronal function. And I've also highlighted UBE3A here. that is the gene that is linked to Angelman syndrome, which is also in the same locus as Prader-Willi syndrome genes as well. So sex-specific imprinted gene differences are more prominent in the embryo head results. Now that we've characterized the lead-induced imprinted gene expression differences in the placenta and embryo individually, let's take a closer look at the bigger picture here. So imprinted genes are a network of genes that are controlled by ICRs, or imprinting control regions, with each ICR containing multiple clusters of imprinted genes, similar to what we saw with the Prader-Willi syndrome, Angelman syndrome example. And there are distinct mechanisms for regulating this cluster of imprinted genes. So our next steps would be to integrate this complex molecular network of imprinted genes by tissue and by sex to identify the maternal fetal functions that contribute to this complex crosstalk, meaning we would have to identify specifically which of these genes are maternally expressed, which of these genes are paternally expressed, which of these genes localize to one region? Are they talking to each other? How are they regulated? So these are the questions, logical next steps to looking at imprinted genes for our ongoing study. So in conclusion, we found that in utero lead exposure is associated with increased placental and embryo weights. Imprinted genes are dysregulated by lead, specifically looking at by tissue and sex. Lead may alter this complex maternal fetal crosstalk as it relates to genomic imprinting. And we need to identify how imprinting clusters are dysregulated by environmental exposures in a very tissue and sex-specific manner to catalog and characterize what's going on, similar to that SNRPN example. So that's kind of what we intend to do. In the near future, our next steps is to extend this characterization to look at genome-wide DNA methylation differences in the placenta and embryo using the same tissues, and looking specifically at these imprinting control regions. Next, we are also planning to conduct hybrid animal experiments, so looking at 2 different strains of mice, which will allow us to really tease apart this allelic contribution that leads to this gene dysregulation. And then lastly, we are also interested in identifying modes of intervention through diet that can alleviate some of these reactive oxygen species, ROSs, in the placenta and embryo to see whether we can kind of rescue the placental weight or embryo weight phenotypes. So these are 3 overlapping projects of future directions that's going on. And a lot of this work is done by a great team of students in the lab. And I'd like to highlight a few students here. There are many that work tirelessly in the lab. Junrupan, who has been helping a lot with the animal exposure studies, as well as the antioxidant in utero studies. Dongyue Wang genotyped hundreds of embryonic samples, and Chloe, who is actually leading the hybrid animal experiments. And I am also thankful for Europe program and the LEED summer research program through EHS for giving these undergraduate students a chance to work on science. And lastly, I also thank my mentors, collaborators, contributors, and our Do Goods Pie research members, and these amazing funding sources, including M-LEEaD, for all of your support. Thank you.
Great. Yeah, excellent presentation. Just for those who are not familiar with what Do Goods Pie means, can you start talking about what that is?
Do Good Spy stands for 4 PIs kind of working in one group led by Dr. Dana Dolanoy. So the Do in Do Good Spy stands for Dolanoy. Good in Do Good Spy stands for Goodrich. S stands for Swoboda, and Pi is, I guess, Perera and Pini, who works on pi RNA. So that's kind of how the Do Good Spy name came into it.
Very fancy name, yeah. Okay, so we have a few questions, but for those who also have any question, please use the Q&A, and then you can add your enter your question. So the first question from the Q&A, and then the finish. So I will, for those who will be watching this video later in the future, so I will be reading the question, but you can also see the question directly from the Q&A or chat. So I'm going to start with the question from Kennedy Clinton. So how did you determine the exposure amount to give to mice?
Yeah, that is a very good question. So the exposure amount is a human relevant dose of exposure. I think it is based on the CDC guidelines, I think about 5 micrograms per deciliter for humans. For the study, we actually measured, we gave the lead in the water, and the amount of lead was measured, the lead that's in the water, and we also measured the blood lead levels in the pregnant dams. So for the perinatal study, the blood lead level for the dams were around 16 to 60 micrograms per deciliter. And for the in utero study, it is the mean concentration was, I think, 9.7 micrograms per deciliter concentration of lead that's in that maternal bloodstream. So those are the measurements for the lead exposure. The
follow-up question could be, so that from the slide, and I see that you are using the like a 32 ppm, which is like a drink water concentration. And then depending on the amount of water each
animal will take, then that will be their blood lead level concentration. Yeah.
But did you assume that just single dose and then comparing the single dose versus the control, or can you talk about are you also considering any dose dependent association of this lead exposure and then imprinted gene dysregulation?
Yeah, that is a very good question, and that's a question that we aim to dig deeper into. We have to start somewhere, but we have to start somewhere in a deeper and a more extensive view. And since I'm just starting my career, I wanted to focus on one dosage based on preliminary data, and we definitely intend to look at lower doses too, because there's, based on the CDC guidelines that's coming out now, there's no safe lower dosage for lead. So we need to kind of look that in deeper, but we intend to do that. But for right now, we just focused on one dosage based on preliminary data, and also to kind of, in order to relate the current data to our previous data, we need to keep some things constant while changing one thing at a time. So I in this study design, I kept the dosage constant to really dig deeper into these tissue-specific and sex-specific effects. But once we learn these effects, then we can go down to a lower exposure level to figure out what is the minimum safe level where we won't see this. I hope that answers your question.
All right, great. So we have several other questions. So the well done, Pini, the sequence data, were placenta and embryo matched?
Yes.
And if you compare the two tissue, placenta vs embryo head?
Yes. So for the study design, I did match the same. So the male and female were from one male and one female per each litter, and each placenta is matched to each embryo head. And Dr. Kelly Bukalski is actually helping me kind of do the statistics to help answer that question, and also to kind of deconvolute the placenta data to figure out cell type specific contribution. So yes, they are matched, and yes, we intend to look at it deeper.
Okay. The next question is great presentation. Please, which reference genome was used in the RNA sequence?
So for the RNA-seq, actually, Dr. Justin Colatino helped me with that. I believe it is MM10, but if you actually give me the email address, I can double check and let you know about which one to make sure.
Yeah, so I cannot see the full name from the this chat. So hopefully we can identify who the person who asked this question later. All right. So another question. I think this is Kelly Bakuski. And then congratulations on a great presentation, Pini. These findings are striking. What do you hypothesize will be the functional consequences of altered expression, this imprinting genes? How might you test those outcomes?
That is a very good question. So there's also a lot of data that I didn't show for this presentation because of the time. I also did pathway analysis, which showed, maybe I can show a couple of slides for, okay. I am gonna share my screen again. So I will, I guess, try to answer the first half of that question. So if you remember the slide from like the loss of imprinted genes and why imprinted genes are important, even though imprinted genes are a subset of genes, they do more than imprinting. It can feed into other pathways, especially growth, hormone regulation, maternal care. So even though it's a small set of genes, it can contribute to a lot of biological pathways. And then that contributes to later life diseases, neurological diseases. For instance, it's Prader-Willi syndrome, Angelman syndrome, but it also can contribute to Alzheimer's disease and other neurological disorders, because I'm interested in the brain, but there's more to it. The second part of the question is looking at like teasing apart why is lead bad at this time point, and how can we figure out why all of these pathways and genes are dysregulated? That is a bigger question that I'm currently trying to answer. I will share my screen very quickly. So this is lead exposure placenta. These are top upregulated genes in the placenta by exposure group. So you can see lysosome, like angiotensin system, adhesion, a lot of these like pathways, type 2 diabetes. So diabetes is linked to embryo, a larger embryo that can also lead to preterm birth. So these are some of the functional implications that can be a consequence of this exposure study. And we can't really answer that now. For that, I would have to like extend this to see like at birth, time the animals, mate the animals, and check their birth time. Are they born before or after? So we can kind of identify some of the functional implications. So these are top upregulated genes. These are top downregulated genes by exposure. So same is true for, I think, embryo head. Oh, this is still placenta. There's embryo head. You can see calcium signaling, oxidative phosphorylation. So that's saying there's a lot of oxidative stress, Parkinson's disease, Alzheimer's disease. So there are a lot of pathways associated with it. So I have a lot of work on my plate to really figure out what this means. So I fully intend to do that, and I can't do that alone. So I have my collaborators and contributors to help me through that. And if you want to, if anybody else wants to kind of sit down and talk about this, I'm happy to do that. I'm very excited about this data. So it's sad for human exposures, but at least we're starting somewhere to figure out what's going wrong.
Okay, great. So it's time, but actually I want to ask one last question before we end this webinar, and because the translation of this finding, I believe, is very important, and then you at the end of the slide, and then you show that so you will be doing like a dietary supplementation, targeting more oxidative stress pathway, so does that mean that you believe that this the imprinted gene dysregulation mainly through the oxidative stress pathway, and then so they try to reverse this lead toxic effect, and then some antioxidant dietary supplement can reverse that this dysregulation process. And but does that mean that this like the current lead exposure in human population, and then how bad or how serious this current lead exposure level that impact this imprinted gene dysregulation, and then any dietary supplementation do you recommend for those who already have high levels of lead exposure, especially for the pregnant women?
So the data is too preliminary to make a big statement like that. But I think the methyl donors, having those folic acid type of methyl supplements are important during pregnancy. It's already recommended by doctors. But the antioxidant diet exposure study is ongoing. We've done the same kind of RNA-seq data with and without antioxidant, with and without lead, to answer that question. It actually has given us more questions than answers. So I will come back and talk when we have that data and I have a better answer for that question. But in terms of translating to humans, I think the main question that we should look at is whether these imprinted genes are also dysregulated in human placenta. And I guess we can't really take human brains out, placenta and like maternal, like cord blood. That would be the next step, and kind of looking at those common genes and focusing on those common genetic pathways and figuring out methods to maybe it is diet, maybe it is antioxidants. I don't know the answer, but that's kind of that would be the next logical step before before I would make a bold statement about, oh, you should take antioxidant rich diet. No, we're still in the process of figuring that out.
Okay, great. So yeah, you already promised that you will be giving another talk next year. Yeah, so we will be looking forward to your next additional experiment and additional this finding, and we really hope to bring you back to this webinar again. But anyway, so thank you very much for your great presentation, and then your Q&A. And thank you everyone for attending this webinar. So we will have a researcher and resident webinar in 2 weeks, and we will see you there. And have a wonderful day. Thank you. Bye-bye.
Thank you.